Thanks for the opportunity to present today. I'm going to be giving a very brief overview of what next-gen sequencing actually is. The intention here is not to go into a tremendous amount of detail. Those questions can be answered by Rick's team, and we have Nick Stanford, one of the senior technological officers here. But instead, it's going to be a clinically focused overview of NGS methods for clinical microbial detection. Just talking through, perfect. So what is NGS? Well, it refers to a collection of high throughput DNA sequencing strategies that can process large amounts of data in a timely fashion. And it's typically in a massively parallel fashion with millions or even billions of sequencing reactions occurring at the same time. And this parallel nature is really what differentiates it from traditional Sanger sequencing. There are two broad types, which I'll discuss very briefly in a bit more detail, but the first is 16S amplicon targeted NGS, which is what's actually done at Microgen. And the second is shotgun metagenomics, which is being done at a group at Mayo and also in Oxford as well, and a couple of Chinese groups. So the first method, which is again used in Texas, targets the 16S ribosomal RNA gene. It encodes the 30S ribosomal subunit, which is specific to prokaryotes. And so it has certain conserved regions, which are shown in blue across all bacteria. And in addition, there are variable regions V1 to V9, which are shown in red, that are specific to individual bacterial species. And so this gene is a starting point for sequencing, as it largely avoids that issue with interference from host eukaryotic DNA, which is present in a typical human clinical sample, while still enabling the taxonomic classification that we need. Also of note, the ITS gene is used for fungal sequencing, but I'm not going to go into that in too much detail. So the 16S gene is then amplified by forward and reverse primers, and then it's pulled and loaded onto a sequencing platform such as the Illumina MiSeq, which is shown on the slide here. And this uses an image-based detection or fluoroscopic detection method, which is known as sequencing by synthesis. So a fluoroscopic change is detected every time a complementary nucleotide, tagged nucleotide is incorporated into the DNA. And then the reads generated are denoised, and then there is a comparison using bioinformatics of the sequenced reads against a curated library of over 50,000 different organisms to generate both species level and genus level distinction. The second method, shotgun metagenomics, is different in that all the genomic material present is sequenced. And so what's not shown in this slide is that prior to this method, host extraction, or host DNA extraction kit is required to remove as much human DNA as possible. This is followed by fragmentation of the DNA present, then sequencing of those fragments, followed by repeated fragmentation, repeated sequencing, until you get contiguous segments that are overlapping. And those overlapping regions are then using computer analytic programs used to assemble sequence reads, which are again compared against a library of known organisms. So why would you use one method over another? Well, 16S targeted sequencing was chosen at Microgen as it's a lower cost. It's also good for lower microbial abundance regions such as the prosthetic joint. And there's also established bioinformatic pipelines available. A majority of the Human Microbiome Project was done with this methodology. And in addition, by targeting the 16S region, you're avoiding largely the issues with host DNA interference. But the trade-off is with the shotgun methodology, you get a better taxonomic resolution at the species level, and it also enables antibiotic resistance and functional understanding, which is not possible on 16S sequencing. And so the projections that you see with 16S sequencing are either based on PCR or projections of antimicrobial resistance based on species identified. So irrespective of which of these two techniques is employed, the general pipeline follows this pathway. So samples are collected, DNA is extracted, and then, for example, for the Illumina MiSeq, the DNA is tagged using custom adapter sequences, and then amplified on a solid surface with covalently attached DNA linkers, and then loaded onto a pipeline such as the Illumina MiSeq, and then reads generated are compared against a curated library to enable taxonomic classification. I know we're running short of time, but I wanted to finish with a summary video just so that everybody is able to see the pipeline pathway on the MiSeq.
Into single strands, and then captured on a solid surface. The attachment occurs through complementary base pairing with oligonucleotides that are fixed to the surface. In the next step, PCR, DNA polymerase synthesizes a second strand of DNA, starting from the annealed oligonucleotide, which serves as a primer. The two long strands are now separated, leaving one strand attached by a covalent bond to a fixed oligonucleotide. The DNA fragment can form a bridge with another fixed oligonucleotide, and another round of PCR proceeds. The strands are separated, followed by additional rounds of PCR. After many rounds, a small patch of DNA forms in the location of each original cellular DNA fragment. These single-stranded DNA molecules represent two sequences that are complementary to each other. Now, one of the strands is clipped at its attached oligonucleotide, leaving molecules of just a single DNA sequence. In this way, tiny islands of DNA form, with each containing many identical copies of a unique DNA sequence. The PCR step produces enough DNA template material for the following sequencing reactions. DNA sequencing begins with annealing a primer to the template. The primer is complementary in sequence to the adapter near the 3' end of the template. The primers provide a 3' hydroxyl group onto which DNA polymerase can begin to add nucleotides. The 4 types of nucleotides have fluorescently labeled groups that can distinguish them. DNA polymerase adds a nucleotide to the end of the primer. The added guanine-bearing nucleotide is complementary to the cytosine-bearing nucleotide in the template DNA. Note that the nucleotides have a blocking group on their 3' ends, so no additional nucleotides can be added at this time. The rest of the nucleotides are washed away. A laser induces the nucleotide to fluoresce, and the color is recorded. Each spot on the solid surface has a particular fluorescence, depending on which nucleotide was incorporated into the growing strand. The fluorescent label and the blocking group are now removed from the nucleotide. Because the blocking group can be removed, this type of nucleotide is called a reversible chain-terminating nucleotide. The nucleotide now has the 3' hydroxyl group required for adding additional nucleotides. More nucleotides are now added, and another becomes incorporated into the growing strand. The free nucleotides are washed away, and the new nucleotide is induced to fluoresce. The cycle can be repeated up to 100 times. The sequences from all the spots are recorded simultaneously. Software packages assemble overlapping sequence fragments into longer pieces, and in this way determine the overall sequence of a genome.
And so once those sequence reads are generated, they're mapped against the curated library, as we mentioned, of those 50,000 microbes. The homology rate is around 99.7% per the lab team. And this is an example of a report that's generated. It gives the species the relative abundance percentage of the entire 16S classifiable reads that are present. And I'd just like to highlight that often this doesn't add up to 100% because it's a proportion of all of the classifiable reads present, and there's a read threshold and also a species detection threshold, perhaps 1%, that's used for clinical reporting. Thank you very much.